Hello everyone, my name is Clan Bear Polar Bear and this is my colleague, Gabriel. And this is our presentation for meiosis cell division. So, have you ever wondered why two siblings look different from each other? Well, that's because of meiosis cell division. So, what's the difference of mitosis and meiosis? Aside from the fact that they have different names, mitosis makes identical body cells. It's also responsible for our growth and repair of damaged cells, but not in meiosis. Meiosis contains genetic variety. It also doesn't make body cells. It makes sperm cells and egg cells. We also know that the human has 46 chromosomes that most of our body cells have. But there's also cells that doesn't have 46 chromosomes. Sperm and egg cell has 23 chromosomes. So, why 23? Well, during copulation, if the sperm cells meet with the egg cells, which both has 23 chromosomes, if we add them up, we get 46 chromosomes in order to form a newly and fertilized egg.
Also called meiosis a reduction division because the starting cell has 46 chromosomes and the last new 4 cells only got 23 chromosomes and 23 chromatids. But before we can make gametes and continue the process of PMAT, the cell division has to go through interphase. First, to make sure that the cells are okay and safe to prevent any harming cells such as cancer cells. So the starting cell has 46 chromosomes and they have to duplicate these chromosomes. Even though the meiosis even start, that also means you're duplicating your own DNA. And since we count the chromosomes by the number of centromeres, so in mitosis, you'd still have 46 chromosomes and 96 chromatids. So in mitosis, you still know the acronym PMAT, P for prophase, M for metaphase, A for anaphase, and T for telophase. But don't worry, you still can use it in meiosis, but PMAT is repeated, unlike mitosis, where we only divide one time. But because meiosis is a reduction cell division, that means that from 46 chromosomes and 96 chromatids until we got 23 chromatids and 23 chromosomes. Now then, let's start with the very first step, prophase 1. We can also call this stage before. In prophase, this where all the chromosomes condense, thickened, and line up with their homologous pair. The word homologous means the chromosomes contain the same size, same germ lines. For example, I genes, they're going to match up. In this stage, the chromosomes are going to do the crossing over, where the chromosomes are exchanging their genetic information. Because of crossing over, it makes up what we called recombinant chromosomes. Recombinant chromosomes is also the reason why sibling looks different from each other, all due to the recombinant chromosomes. Metaphase 1, M for middle. All chromosomes are going to be in the middle of the cell. It's also different from mitosis because all the chromosomes are going to be in pair. Microtubes from opposite poles of a spindle. The spindle is going to attach all homologous chromosomes. Anaphase 1, A for away, because all the chromosomes are going to be pulled by different poles by the spindle. And last, telophase 1, T for 2, where in this phase where it formed only 2 nuclei, and because of meiosis 1 is complete, we successfully divided the chromosomes from 46. But we're not done yet, because we have to divide the chromatid to 23, and that's how the next phase will begin.
Prophase 2. The events in prophase 1 won't be repeated, like the crossing over, but the spindle will now start to form, like in prophase 1. And once again, the chromosome became compact, and the nuclear membrane disappears by the end of prophase 2. Metaphase 2. In this phase, all the chromosomes are in one single straight line. They're also not in pair, unlike metaphase 1. Anaphase 2. In this phase, if we remember correctly, A is for away, but this time, the spindle fibers are not pulling away the chromosomes, they're pulling away the chromatids instead. Telophase 2, you can see the nuclei is reforming, and now we had 4 new cells. And meiosis cell division is now complete. But what if there's an error to the cell division? Like for example, one of the chromatids didn't separate correctly. We call this non-disjunction. This can also lead to genetic disorder. Like for example, Down syndrome, trisomy 18, and more. Oh, that's all for today's video. Stay safe and stay healthy. Bye!
Bye.